Welcome to IBDP Biology Unit B1.1, Carbohydrates and Lipids, where we explore the molecular basis of life. Carbon atoms form the foundation of all biological molecules, capable of creating 4 covalent bonds with other atoms. Let's start by understanding why carbon is so special in biology. Carbon can form 4 covalent bonds by sharing electrons with other atoms, making it incredibly versatile. Here we see a carbon atom at the center forming 4 covalent bonds, sharing electron pairs with 4 other atoms. A simple example is methane, where one carbon atom bonds with 4 hydrogen atoms. Carbon compounds come in 4 main structural types. Carbon compounds exist as branched chains, unbranched chains, single rings, and multiple ring structures. Now let's explore how small molecules join to form larger ones. Condensation reactions join small monomers together to create large polymers, releasing water in the process. 2 monomers combine through condensation, forming a polymer and releasing a water molecule. Monosaccharides are the simplest carbohydrates. Monosaccharides are single sugar molecules containing either 5 carbons, called pentoses, or 6 carbons, called hexoses. Here's glucose, a hexose sugar with 6 carbon atoms arranged in a ring structure, the most important energy source for cells. Polysaccharides are large polymers made from many sugar monomers. Animals store energy as glycogen in liver and muscles, while plants store energy as starch in roots and seeds. Polysaccharides consist of many glucose units linked together in long chains for efficient energy storage. Plant starch exists in 2 forms: amylose with unbranched chains and amylopectin with branched chains. Some polysaccharides provide structural support rather than energy. Cellulose is a structural polysaccharide that provides strength and rigidity to plant cell walls, making plants stand upright. Cellulose forms strong parallel fibers that are cross-linked, creating a rigid structure perfect for plant support. Lipids are a diverse group of hydrophobic molecules essential for life. Lipids include oils, fats, waxes, and steroids, all sharing the property of being hydrophobic or water-repelling. The 4 main types of lipids each have unique structures and functions in living organisms. Triglycerides are the main form of stored energy in animals. Triglycerides consist of 3 fatty acid chains, bonded to a single glycerol backbone through condensation reactions. A triglyceride molecule has a glycerol head with 3 long fatty acid tails, perfect for compact energy storage. Triglycerides are stored as adipose tissue, providing animals with long-term energy reserves and insulation. Phospholipids are the key structural components of cell membranes. Phospholipids have 2 fatty acid chains, a glycerol backbone, and a phosphate group, making them amphipathic. A phospholipid has a hydrophilic phosphate head that loves water and 2 hydrophobic fatty acid tails that repel water. This amphipathic property allows phospholipids to form bilayers in water, creating cell membranes. Phospholipids spontaneously form bilayers in aqueous environments. In water, phospholipids arrange into bilayers with hydrophobic tails facing inward and hydrophilic heads facing outward toward water. These bilayers form the fundamental structure of cell membranes, separating the cell interior from the external environment. Carbohydrates attached to proteins enable cells to recognize each other. The ABO blood group system uses glycoproteins on red blood cell surfaces for immune recognition and blood typing. Glycoproteins extend from the cell surface like molecular markers, allowing cells to identify friend from foe. Let's review the essential concepts from carbohydrates and lipids. Carbon's ability to form 4 bonds enables the vast diversity of biological molecules. Condensation reactions join small building blocks into large macromolecules essential for life. Carbohydrates serve dual roles as energy storage molecules and structural components in organisms. Lipids provide long-term energy storage, form cell membranes, and function as signaling molecules. Understanding molecular structure is key to understanding how biological molecules perform their specific functions in living systems.